And that, of course, brings an issue of antimicrobial resistance genes and the transfer of those genes. The question is, do we also within those biofilms then have a variety of species? Are they remembering inside pathogens? Are there exchanges of resistance genes and of virulence genes of those pathogenic and non-pathogenic species? And what does it eventually do? To understand that, we have performed ourselves a large study making an incubation of microplastics in a variety of water environments, being like a kind of a harbor for the kind of small family boats and the sport boats, and then the industrial harbor where the big industrial activities were happening, aquaculture site, and then also countryside canals and sluices for the water which has been used for different urban and agricultural purposes. About 1 meter below the surface, we have placed specific cages which contain the polypropylene microplastics of size up to between 25 and 85 micrometers. We use the glass bits as sort of a control, and we compare then what was microbiologically happening on them in comparison to the water which surrounded them. And we have basically seen that a significant accumulation of microorganisms on the particles was being formed, creating a very dense and diversified structures. That was saying that actually dependent on the location, depending on the activity, a very different microflora has been actually formed. When we analyzed using the sequencing these microflora, we have noticed that the biofilm communities, shown here, which were associated to microplastics, were independent ecological niches when were compared to the water where those plastics were incubated. So that means that the microplastics, or in general, maybe physical structures, were creating a place where the microbes could attach, adhere, colonize, and behave somewhat differently than they would behave in the water itself. Among the species which we found on the plastics, we saw Salmonella, different Vibrios, Haemophilus, and Aeromonas. And these were just some of those which were really found to be both human and fish potential pathogens. When we looked into the genetic material placed within those biomass, we saw a lot of virulence genes and a lot of antimicrobial